In this fourth part of vision, I'm going to get into the brain, and we're going to talk about the visual pathways through the brain to the primary visual cortex, which is our first higher level, so cortical area for processing vision. We usually call it a visual receiving area. And then we're going to talk about the organization of V1, which is the primary visual cortex. So as I often do, I'm going to start with talking from these pictures, and then I'm going to repeat a lot of it with the verbiage, with the words on the slides, so that for me it helps. I think it helps a lot to see it, but then also for taking notes and everything, it's nice to have the words. So if we're looking at the left picture there, that we have that information going to the retina and back through, so that is the optic nerve leaving the retina, going back to the lateral geniculate nucleus and to the superior colliculus, which are both kind of pointed out there, although very tiny. And so we have to cross. Some of that information is crossing at the optic chiasm, though some of it's going back ipsilaterally. And from the lateral geniculate nucleus of the thalamus, we're going to take that information that's going to be sent to the visual cortex. And some of that is more clear in the picture on the right, although the superior colliculus is left out of this one. So that's that area in the midbrain that is allowing us to take in information from various modalities so that we know where we want to move our eyes. So the picture on the right, when we're looking straight forward, this is going to do the visual field stuff and talk about contralaterality. Again, I know we've done this, but I feel like it has to be done with vision for, I don't know, I feel like it has to be done with vision. So because it does have this complicated contralaterality, it's of the visual field information. So if I'm looking straight forward, right between, so fixation cross or whatever, between the heart and the smiley face, what's happening at the eye, right, with the way the lens and the cornea are refracting and bending that information is back on the retina. What's in the left visual field is going to be over on the right half of each retina. What's in the right visual field, so the smiley face is going to be hitting the left half of each retina, and of course it's an inverted image. And so those parts of the eye, and I'm just going to talk from the right eye, and you just can know that the same thing's going on with the left eye. So if I'm going to talk from the right eye, the right part of the right eye, in this case, it's receiving the information about the heart, the upside down heart now. That's what's on the retina. That is the part that's closest to the temples, and you can see that with the red lines that's going back ipsilaterally, that stays on the same side, whereas the left part of the right eye, so that part that's closest to the nose, that part is going to cross over, and that's crossing over is happening at the optic chiasm. Okay, and so we're going to shuffle that information in together. So the right part of the right eye and the right part of the left eye, both of which are receiving slightly, so 6 centimeters apart, but basically the information from the left visual field, those are going to, the left eye is going to cross over because it's the one closest to the nose now, and that's going to be shuffled together appropriately, so we match up what's appropriate with each other at the lateral geniculate nucleus. And the lateral geniculate nucleus is the thalamic nucleus doing the organizing, summarizing, filtering information, or receiving top-down information so that it kind of knows some of what it's expected out there, and then sending most of that information on to the primary visual cortex. So I'm going to walk you through with the language, starting with those subcortical regions that we've taken the information out from the eye back through the optic nerve and gotten it. It's going to synapse first in the lateral geniculate nucleus, often shortened to LGN. This is in the thalamus. This is where the majority of the signals go. And it is organized in these 6 distinct stacked layers that I'm going to show in a second here. And it is going to keep the input from each eye separate from each other. The other subcortical region that we're going to talk about now is the superior colliculus. So approximately 10% of these signals go to the superior colliculus in the tectum. Some really small percentage also go. So different kinds of retinal ganglion cells than what we've talked about so far also go to an area of the hypothalamus that is going to tell us about what light levels we're in, and it's going to affect our sleep-wake cycles. So we're going to talk about those when we talk about sleep-wake cycles, but for now, we're going to ignore them. Okay, so I'm going to walk us through those two subcortical structures. First, the lateral geniculate nucleus, or LGN, of the thalamus, and it is doing what those thalamic nuclei are doing. The ones that are receiving sensory information are controlling the flow of the information, organizing the information, filtering and summarizing the information. I usually talk about them as the librarians, so that they're organizing information so that we can, in this case, see what's relevant given the scene. We're getting much more input than we are sending output. The output is going primarily to V1, or the primary visual cortex. We're getting input from many places, at least, including definitely, we know, the retina, right? Because that's where the vision is coming in from the eye, but also from other nuclei in the thalamus, from within the lateral geniculate nucleus itself, so communicating within myself, from the cortex. So if you think about bottom-up versus top-down processing a bit, and how these expectations and background knowledge influence what we actually see in the environment, we see these white matter tracts going from the cortex back down to the lateral geniculate nucleus that is going to influence how it's going to organize and summarize that information. And then also from the brainstem, so that really lower level attentional information of if I hear some kind of noise out there, that specific noise might tell me something of what I'm looking for, and especially if it's something jarring that grabs my attention, that's going to have an input to the lateral geniculate nucleus. Okay, and again, the output is primarily to the cortex, to the primary visual cortex, and we see this 4-to-1 ratio of afferents to efferents. So again, the afferents are arriving fibers, so 4 arriving fibers for every 1 exiting or efferent fiber going to the primary visual cortex. And what's being shown in the picture to the lower left is, so you can see the thalamus and being distinguished, and then there's a little square around the lateral geniculate nucleus. So notice it is lateral, and it looks like a little knee, which is where it gets its name, geniculate, and it is a nucleus of the thalamus. And then that little square is magnified so that we can see the 6 layers, those 6 stacked layers that are receiving, that different layers are receiving input from different eyes. So layers 1, 4, and 6 are receiving input from the contralateral eye, and layers 2, 3, and 5 are receiving input from the ipsilateral eye. And if you think about how this works, so if information is in my left visual field, it's hitting the right sides of each retina. Some of it's crossing over at the optic chiasm, and it's getting shuffled together into the lateral geniculate nucleus, such that if I took an electrode at some place in that lateral geniculate nucleus and went down through, so perpendicular, that every place in that column is going to be responding to a particular location in space. It has this retinotopic map, and these layers that are getting information from the different eyes are shuffled together so that that information lines up with each other in this kind of miracle of neural development when our brain is developing. The other thing that is also being shown here is that we have those 2 magnocellular layers, so layers 1 and 2, and those 4 parvocellular layers, layers 3, 4, 5, and 6. I skipped some of this from the book because we were a little bit behind, and I think it's okay for how we're moving forward. But the magnocellular layers are mostly responding, so they have these large receptive fields, and they tell us about movement in our visual world. And the parvocellular layers are receiving input from those cones, and they are telling us about the little pieces and parts of objects so that we can put objects together, really. And then if you notice, there's a white space between those layers, and the author's pointing to that, saying those are coniocellular layers. When I was in undergraduate taking this class, they did not talk about the coniocellular layers at all that I remember. And so when this got added, I had to look into it a little bit. They still don't know completely what those coniocellular layers are doing, but it looks like, if I remember correctly, from what I understand, that these are receiving information from the short wavelength cones. Okay, and the superior colliculus, as you remember in the tectum, it receives approximately 10% of the signals, and it is controlling eye movements and other visual behaviors. So its function is to localize things in space, use that for eye movement control, but it's also receiving information from multiple modalities, because again, if we hear some kind of noise, we're going to want to move our eyes there. If we hear a noise, feel a touch, see something move, and all of that's happening together, we're going to definitely want to move our eyes, even more strongly want to move our eyes there. So it has these spatiotopic maps, and also it allows for multimodal spatial coordination. I talk about the superior colliculus much more in my sensation and perception class, as it is one of these areas that we don't understand very much, and they are still doing a lot of research trying to figure out exactly how we figure out how to move our eyes. And this is one of the real catches of what makes it very difficult to create perceiving machines, because we can't get them to move their eyes the way that we move our eyes based on various pieces of information in our environment. Okay, and again, the information goes from the lateral geniculate nucleus, so it is the thalamus that is sending that information on to the primary visual cortex. And I'm saying here, and I may have already started alluding to or using these terms interchangeably, as they do get used interchangeably in the field: primary visual cortex, striate cortex, which means striped, it has a striped appearance, or V1, those get used interchangeably, and they all mean primary visual cortex. Okay, we have pattern recognition in the cerebral cortex that occurs in a few places. We're going to start with the primary visual cortex, that is receiving information from the LGN, and it's responsible for this first stage of cortical visual processing. And we've already discussed it very briefly back when we discussed blind sight, because some people with damage to V1 show this blind sight. Wherever that damage is in V1, that part of their visual field, they are denying that they see anything there, but they have an ability to respond to those visual stimuli. Okay, so we've gotten this information to the primary visual cortex, also called the striate cortex, and I'm going to talk about a number of things there. One, feature detectors. So we've already talked about receptive fields. These are neurons that are responding to certain aspects of the environment. And then we're going to move into the cortical mapping, or the organization of the striate cortex, where we're going to see this correlation of the location of neural activity in the striate cortex and the visual cortex with the position of an object in the visual field, which is a retinotopic map, as I'm going to call it. You can see the word retina in there, because remember, what's happening with vision is that we have the light information out in the world that is being reflected off of objects. It's going into our, it's being reflected into our retinas, and we're going to have this upside down reversed image that is maintaining that information about the position of the object in the visual field. That's on the retina. That information from the retina is maintained in a retinotopic map in the lateral geniculate nucleus, and we see the same kind of retinotopic map in the striate cortex. I'm also going to discuss the cortical columns and hypercolumns as part of this mapping, or how the visual cortex is organized. And very briefly, I'm going to talk about cytochrome oxidase blobs, which are our color blobs, our blobs of color. So I'm going to start by talking about those receptive fields. So what the neurons are responding to in the striate cortex. And I'm just going to talk about the feature detectors. There are different neurons responding to different things, as we're going to talk about color blobs in a moment. But we have our feature detectors are responding to features in the environment. We have simple cells responding to the orientation of a line, whether it's vertical, oblique, or horizontal. Complex cells are responding to a particular orientation of line, plus the direction of movement is important for them to respond. And then hypercomplex cells are what are sometimes called end-stopped cells, because they're really reading the ends of those lines and angles and so forth, but they're responding to the length of the line, the angle. So if we have something that has a right angle or a 45-degree angle with the line next to it, and as well as the direction of movement. And it was David Hubel and Torsten Wiesel who told us about these feature detectors. They actually were also the ones who were recording from the retinal ganglion cells earlier when we were looking at the cat that was looking at the screen. And in 1959 and 1965, they discovered these feature detectors for which they won the Nobel Prize in physiology and or medicine. If you look at the picture, so what that's showing is, so remember we were at little dots on the screen. So our stimuli with those center surround receptive fields, what those neurons were responding to best, right, was a dot of a certain size hitting those neurons. And as they got out into the visual cortex, you can see from the picture that that was still the stimuli that they were using. But here it's showing that one day what happened was they had the cat hooked up to the machinery and everything, and the slide got stuck so that it ended up showing just the horizontal line. And actually, I think it was the movement of the horizontal line coming down. And that's when the neuron fired. And David Hubel is quoted in that Nobel Prize winning speech as saying, the neuron fired like a machine gun. They were so excited, and of course they changed all their stimuli after this serendipitous result to see that what's happening at the level of the visual cortex is that we are responding to, the neurons are responding to features of our environment, no longer just responding to the little dots that are being put back together to make up those features. And so I quickly went through what each kind of cortical cell, what these different feature detectors are responding to, but I'm going to do this with pictures, as I often do, as you may have noticed. So this is showing an example of a simple cortical cell that is responding to a vertical line or vertical bar, so that orientation. And you can see that it is excitatory. We still have that excitatory center in this case and an inhibitory surround, where if I'm a vertical bar hitting right at that excitatory center, we're going to see so what's happening in these different figures is the vertical bar in the middle figure is where we see the neuron firing quickly. And what's being shown over on the right is the number of impulses per second. So vertical for this simple cortical cell is at 0 degrees because that's what I'm responding to. And then as we move off of what I'm responding to with these different levels of oblique, whether that's a 20-degree off or 40-degree off, we see that that neuron is going to respond less and less. So if we go back to the middle picture, I'm at this kind of oblique line, and if you kind of mentally put that over the picture on the far left, you can see that that oblique line is going to be hitting mostly excitatory space, but also hitting some of that inhibitory space. So we're going to see less firing for this particular neuron. Okay, there's going to be a neuron that responds to that degree of oblique line. It's going to have a different pattern, but this particular neuron is going to have less firing than if it's a vertical line hitting in the middle of that space. I have in here this measuring the receptive field of a simple cell in the visual cortex. I'm going to recommend that you watch it. I don't require it, but I usually show it in class for about 2 minutes. And it's that we're measuring the simple cell, the response of a simple cell, and that noise in the video is the neuron firing. It's really quite cool to see how they did this. It's about 11 minutes long, and really the first 2.5 minutes is usually what I show. But if you want to go somewhere in the middle, they're also measure the receptive field of complex cell. I've had people ask me if I've watched the whole thing. It's only 11 minutes, and I say no, I haven't. And so these guys did win the Nobel Prize in physiology and or medicine, and you just have to realize that they did not just spend this 10 or 11 minutes, right? They spent hours and hours and hours, days and days, and weeks, months and years of their lives telling us about these feature detectors and what neurons are responding to in these different areas of the brain and the visual system. So that was the simple cell. More briefly, I'm going to go through the complex cells and the hypercomplex cells, or what are sometimes called end-stop cells. And for the complex cells, this is being shown in A. And this particular complex cell, remember we have many of these responding to different features, different orientations, different directions, but this one is responding best to what's being asterisked there, the third one down. So that particular orientation of line. And what you'll notice in what it's trying to show there in the firing pattern is it's when it's moving up and to the right that the neuron fires. When it's moving down and to the left, the neuron does not fire. Moves up and to the right, the neuron fires. Down and to the left, it's not firing. Okay, so this particular complex cell is responding best to that specific orientation of line moving up and to the right in that particular direction. And you can see above that, if we get a little bit off, that there might be some neural firing, but the more off we get, remember we're going to be hitting some of that inhibitory space and there's going to be less neural firing. The hypercomplex cell is being shown in B, and in this one, you can see the second one is what's being responded to the best. So it's what that shadow is trying to show you is that it's the receptive field, or part of what the hypercomplex cell is responding to is that particular length of the line. And in this case, it's a right angle, and it's when it's moving up and to the left. Okay, when that particular length with that particular angle of that orientation of line is moving up and to the left, the neuron fires. Down and to the right, no firing. And as we get a shorter line, so what's above it, we see some neural firing, but not as much. If we get a longer line, so what's down below it in the picture, we again, when it's moving up and to the left, I get a tiny bit of neural firing, not when it's moving down and to the right, but not the same as when we have that specific length of line. So we do want to see the ends of objects and what direction they are moving for exact motion information. And so I had some of this earlier, but I'm just repeating because I think receptive fields are really important. Neurons are now in the visual system are responding not just to light, but they respond to some patterns of light and not to other patterns of light. And as we're moving farther from the retina, those neurons are responding to more complex stimuli. As we're in the striate cortex or V1, those neurons are still just responding to features of the environment, which is how they got this name of feature detectors. But remember, we're still pulling this apart into features. We need to put it back together, right? I want to see tables and chairs and animals and people. So we need to send this on for higher level processing. Okay, so the organization of the striate cortex, or the mapping of the striate cortex, is, again, it's retinotopic. So I'm representing the visual space on the retina, and that retinotopic map is being maintained to the primary visual cortex, which is how we talked about blind sight at the very beginning of the class, where if I have damage to my lower left visual cortex, I'm going to be missing the upper right part of my visual field. We also have cortical magnification of the fovea, and what this means is that the foveal vision is magnified. It has more representation in the cortex, because remember, that's where my detailed vision is, and there's not so much detailed vision of the periphery, so we need to have more representation of that fovea in the visual cortex. In addition, we have these hypercolumns, okay, that are made up of various columns, and that's what's being shown in the pictures that I'm going to talk about here for a second. So you'll notice on the right hand image there's an L and an R and an L and an R. Okay, well those are two location columns next to each other, where the location column is location on the retina, because remember I have to represent the space on the retina where that light information is being processed. And the L and the R are showing these two different ocular dominance columns that are going to be part of what makes up those location columns, where some of my neurons are responding best to the left eye and some of my neurons are responding best to the right eye. And in addition, we have orientation columns coming sort of the other way. So those simple cells that are responding to particular orientations of line, we're going to, if I have a vertical line in my right visual field up at the top, I'm going to have a location column, or I'm going to have a hypercolumn that is representing a particular location. Down in my left lower visual cortex, that is going to be responding to that particular orientation of line. And if that orientation of line changes, you can see kind of over on the left that I'm going to have that same location column is going to be responding, but it's going to be a different orientation column within that to represent a different orientation of line. The other thing that is represented in here are these cytochrome oxidase blobs, and this author has them as going down through, so you can see the blobs are labeled, and they're those cylinder looking things. It has them going through the entire column. My understanding is they are the fourth layer, is the cytochrome oxidase layer. And this is just, that's a, cytochrome oxidase is an enzyme that is showing a high metabolic activity. These are our color blobs. This is where we are responding to color. So again, we've pulled this apart, this visual information apart into features. Those orientations of line, movement, length of line and angle and movement, the direction of movement, and color is separate from that in the visual cortex. Now we really want to put this back together so that we can represent the world in a meaningful way.